Hello and welcome to Psyched, the show where we explore psychedelics through social, economic, and political perspectives. Next up, we have Ian Mcdonald. Ian is responsible for developing the overall strategic vision, as well as assembling the intellectual and capital resources to bring scientific innovations to market. Prior to founding Bright Minds, Ian worked in a senior role at a Canadian investment bank and in private equity in Vancouver, London, and Toronto. He maintained deep relationships with investors and helped develop capital market strategies for issuers. Under Mcdonald's guidance, clients raised hundreds of millions of dollars in capital. Ian has served as a member of the board of directors of several TSX Venture Exchange, Canadian Securities Exchange-listed, and private companies. Ian, thank you so much for joining us and welcome to Psyched.
Merrick, thanks for having us. It is a pleasure to be on here. That was a great introduction. Just to give you guys a little bit more detail about Bright Minds, it was something that I got interested in 2015, saw the potential that psychedelics had on a wide array of disorders, but primarily psychiatric disorders. And with that, I started doing a deep dive in the literature and really understanding where the problems were today and try to figure out the next 25 years of psychedelic medicine, and came to the conclusion that while these drugs are a great initial starting point, they can be tweaked and modified to fit specific, tailored to specific indications. So I set out to find the smartest guys and most capable psychedelic drug designers I possibly could. And I think we've assembled quite a team. So unfortunately, they weren't able to join me today due to conflicting schedules, but I have the next best thing, which is a recording of myself and my two scientific co-founders, Gideon Shapiro and Alan Kozakowski, which I will share right now, and then stick around for questions after.
When you screen share, yep. Okay, I think you're good.
A little bit about yourself.
Sure. So I started my career as a medicinal chemist in Sandoz Pharmaceutical in Switzerland back in 1986, and have been involved basically in CNS drug research ever since that time. So a nice tradition where Albert Hofmann, who invented LSD, used to lead the company's research department. Since then, basically, after being involved with Sandoz, I moved on to become an entrepreneur, and most recently have been very excited by opportunities I've worked on, this new emerging space of new drugs, especially for psychiatry, which originated with the use of ketamine to treat basically depression is the biggest breakthrough in psychiatry in 50 years. That has this novel mechanism related to psychedelics where you get a brain reset, sort of what we call a chemical electroshock. So I got really excited then when Ian came to me in this new emerging field of psychedelics, which really is very parallel to what's now already been demonstrated with ketamine and the second generations of drug that I've been working on as part of a biopharmaceutical, my latest involvement. So this has really been tremendously exciting because it's a parallel that could be even a bigger breakthrough than the ketamine and the NMDA field, where basically these first generation drugs, similar to ketamine, have problems and side effects and are really ripe for creating a second generation of safer drugs to basically treat a greater population of patients and diseases.
Thanks. Alan, do you want to tell us a little bit about yourself?
Okay, well, like Gideon, I was trained as an organic chemist. my PhD at Berkeley, did postdoctoral studies with a Nobel Prize winner, E.J. Corey, at Harvard, then went on to start my own career at the University of Pittsburgh and a number of other universities, such as Georgetown University Medical Center. So I worked as a professor for more than 40 years, and published, 500 some publications, hundreds of patents, or 100 plus patents. And during that time, also did a sabbatical in neuroscience, so I became very familiar with brain neurochemistry. Last year, Ian came to me and encouraged me to work with him on Bright Minds Biosciences, which I was happy to do because this really fell into the particular niche of medicinal chemistry and neuroscience that I've been interested in for years. We worked on things like huperzine A, which is actually now sold in stores like GNC, Walgreens, and CVS Pharmacy. It's a memory restorative agent that came out of Chinese folk medicine. It's actually isolated from a club moss, and it's a cholinesterase inhibitor that's quite an interesting molecule. We also worked on things like cocaine, trying to find antagonists, likewise angel dust and PCP, and actually started a company, Actuate Therapeutics, where we developed some glycogen synthase kinase 3 inhibitors, which are now in the clinic in phase 2 clinical studies for cancer. So very broad background in medicinal chemistry and neuroscience. And a particular interest with regard to Bright Minds was the fact that we had worked on 5-HT2C receptor ligands with a famous neuropharmacologist at University of North Carolina, Brian Roth. And that's interesting because psilocybin, which is isolated from these magic mushrooms, has 5-HT2C activity, but more important, it also interacts with another of the serotonin subtypes, the 5-HT2A receptor. So this has given me an opportunity to make use of the knowledge I've gained from those past studies with Brian Roth in order to push Bright Minds forward into some new arenas in terms of re-engineering psilocybin to make it even something better that could be used to treat a host of medical indications.
And I think that's an important point you touched upon with your work with Brian. We have now actually licensed the product of that 3-year plus program there, and I think over $4 million spent in total.
Right. That was an NIH-sponsored research grant. It was actually a 4-year grant that had been awarded for us to look at developing new 5-HT2C ligands, in particular for application to schizophrenia. But of course, if you got good ligands at that site, you can use it for other indications such as obesity. And the interesting aspect of that work is that in that process of engineering very selective 5-HT2C ligands, we also discovered a number of ligands with the 5-HT2A activity without the more dangerous or unwanted 5-HT2B receptor activity, which is responsible for heart valve disease.
Hey, Gideon, do you want to just go into a little bit more detail on the science, the 2B activity that we are eliminating, as well as the pharmacokinetics, and why that's important?
Sure. So the object of our drug design essentially is that all of the first generation psychedelics, LSD, psilocybin, et cetera, have very potent activity at 5-HT2B receptors that are located on the heart valves and are very strongly associated with valvulopathy and heart damage. The psychedelic effect and the therapeutic effects are all located in the 5-HT2A. So essentially, there's a major problem with getting regulatory approval and scrutiny for this intrinsic toxicity with the first generation drugs. So as Alan said, our goal as essential as medicinal chemists is to engineer out that 5-HT2B activity, which we typically call off-target as a side effect, and to have selectivity for the subtype 2A, which mediates the therapeutic effects. So I think one of the issues is people are a little underplaying this. Basically, to date, psychedelic drugs can be dosed once, and that's the problem as we get into the approval process, and beyond just the simple fact that they work in all these diseases, those liabilities are basically going to circumscribe the application. It's pretty much a given in our mind. So for broader use and safety, clearly a second generation drug that is selective for the 5-HT receptor as a medicinal drug is better. And then on the second point, which you touched on, the duration of action is also rather critical to clinical use and to eventual use in an at-home setting. So to move in that direction, we really are trying to engineer that duration of action so that the trip experience is basically not too long, is long enough to be effective, but doesn't have a duration that's prohibitive for treating a patient, either in a modest clinical setting like a therapist setting, or even hopefully at home. So LSD clearly, a trip time of over 10 hours is really going to be problematic for its application to the general population. And psilocybin is a little better, but still it's up to 6 hours. So typically for ketamine, people can't get released from the hospital, and it has to be given in a hospital setting until they're ready to basically go home and that their function is good. So that's the problem. I think we're going to have the same thing with this first generation of psychedelics that people have with ketamine, only worse. So if we can make the drug safe, make the duration of action manageable for a short session of therapy, and hopefully for home use, it would be a tremendous breakthrough for patients generally.
And I think there's a question of why this is happening now as opposed to in the 1960s, and I'll just quickly address that. Today, thanks to advances in technology, we have far greater insight than any other point in history into the mechanism of action. That knowledge allows us to create targeted therapies like Gideon was just describing. And with that, we really feel that we're entering the golden age of psychedelic-based medicines. So if you go back to that previous period of, call it psychedelics 1.0, the 1960s, where LSD was invented, where psilocybin was popularized, and some of these other compounds started being created, there wasn't anything that was really known about the receptors when they started research. They just knew that there were these things called receptors and the molecules bound to them. It was only in the 1990s that we were able to identify the, I believe, 14 different receptor sites and begin to understand how they work to control mood. So we still weren't sure about what those receptor sites look like. And for those out there, it's basically a lock and key model. The receptor is the lock and the molecule is the key. So you're trying to fit that in perfectly. Which is what Gideon and Alan are pros at. But that really required advances in something called protein crystallography, which is being able to identify what that structure is, what the lock looks like. Also things like functional magnetic resonance, which is brain imaging, really gave you an idea of how these drugs worked in the brain. Clearly they light up, they help with communication within the brain and light up a number of areas that wouldn't otherwise be activated. Also, we didn't have the computational power to analyze all this data and create superior molecules. And this is only a relatively recent advance. So maybe, Alan, you could touch on the computational chemistry aspect and our work with Guido Vistoli.
Sure. What's happened in the last couple of years, and this is work done in part by Brian Roth, is that X-ray crystallography has been carried out on the 5-HT2A, B, and C receptors. So the X-ray crystal structures are available of these proteins, these receptors, bound to small molecules. And so that information can be used to engineer new compounds. The drug designers can get to work like Gideon, and we can look at the small molecule bound to this receptor, the macromolecule, on the computer screen, see where the interactions are of small molecule to big molecule, improve those, change those, alter those so that the ligands become, that is the small molecule, the drug becomes more selective for one of the receptor subtypes over the other. And the molecular modeling has been done at the University of Milan by a professor in the Department of Chemistry, Giulio Bistoli. So he's a super modeler. He's got automated algorithms that allow him to dock these small molecules and find the best binding site of small molecular drug to the protein receptor. So I think that summarizes that fairly well. So, those X-ray structures you can acquire, they're freely available to anybody or through the protein data banks websites.
Mm. And I think I would also point out that the assays are just as critical. So now we also have a leading pharmacologist with John McCorvey, who came out of Brian Ross' lab in understanding the function and the transduction of signals for these various receptors. So that is also a key piece that we're only just now really starting to understand how different psychedelic molecules differ, not just in the receptor they bind to, the specific subtype of the human clone, but how they create signals in nerves and neurons.
And could you just explain that briefly for the non-scientific folks that are listening?
Yeah, so basically nowadays with molecular cloning, you're essentially able to isolate the DNA clone of the human receptor itself that's not in the brain and express it in a cell, a mammalian cell, and then measure a signal. Typically, that signal is an ion current that you transfer into a fluorescent signal and look at what's called signal transduction. So when the serotonin is released in the brain and binds to the postsynaptic receptor, how does it create a nerve signal and an impulse? And that's really what it's all about, being able to do that efficiently in a testing manner to basically design drugs that are selective for certain signals versus others. So that really is a key development, to have that assay technology and deploy it really in a efficient fashion to find a drug. And that's what we've set up now with this collaboration with the University of Wisconsin, a leading laboratory.
And from a commercial standpoint, I think it's important to point out that in order to justify the massive expense of regulatory quality clinical trials, you need to have strong intellectual property. Unfortunately, the first generation compounds like psilocybin and like LSD, that's pretty much impossible. When you modify the compounds to create superior drugs, the patent office will reward you for that innovation. It's these second generation molecules which will not only revolutionize the treatment of a range of disorders, but also create shareholder returns that can further be reinvested into research and development, creating, driving innovation into the psychedelics field. And that's probably a good point for you to touch upon, Gideon, with your tremendous experience in not only writing, but defending patents and knowing how this works from both the big pharma and small pharma side, what they're interested in and why they're interested in the intellectual property.
Sure. So the gold standard for a pharmaceutical and for patent protection is what's called a novel composition of matter. That's essentially the same thing you'll hear it called a new chemical entity. That means it's a molecular structure that has not been basically described to date. So psilocybin, as you said, and all the natural products are natural products where you can't have. They're not novel compositions. They're now well known for many decades. So we as drug designers, the advantage is that when we design a new molecule, essentially we're able to create a new chemical entity that has a new chemical structure at the same time that we're designing it to have less side effects and to be a superior selective drug. So they go hand in hand, fortunately, and that really is what you really need for the payback on that huge investment of up to $100 million or more to take a drug to market. So Unfortunately, in our capitalist society, that's still really necessary for us to bring medicine to patients. We do have to be able to pay for it some way, and to date, and also to be able to partner with large pharma, because it's also very valuable to be able to have a partnering with a large pharmaceutical company to help you get through the very late stage expensive clinical trials. So without having that, it's very difficult to do commercial partnering with pharmaceutical companies, which really is going to be the ultimate key to deploying these broadly, really for large patient populations and general use safely. So that's really the key to what we're doing. Unfortunately, Alan's invention is really a great start, because this patent has already been issued. It's already been granted in the United States. So essentially, we start with a portfolio of compounds that is patent protected for composition and is being globally prosecuted. And then as follow-on, we have a novel portfolio of other novel chemical entities, patents that we have filed. So we have a pretty broad estate. I think the best in the business in psychedelics currently, and we're really optimistic at being able to carry this through.
And again, that's something that we plan on continuing to build. We certainly have the team and capability to do that. I would say, agree with Gideon. I think that we're best in class in terms of intellectual capital on the drug discovery side. It's probably worth touching on our partnerships with clinicians to drive our development program. So once those molecules are discovered and characterized in animal models, we then take them into the clinic to test in humans. Our primary indication at the moment will be a disorder called cluster headache. We have signed on the lead doctor at the Mayo Clinic who specializes in cluster headache. So maybe Alan, you can just touch a little bit on how that generally works with things like that transition into from preclinical to clinical.
Well, that involves a fair amount of jumping over hurdles and barriers in terms of carrying out the preclinical work, which we generally abbreviate as ADMET, which is absorption, distribution, metabolism, excretion, and toxicology. So one does, some key studies which are, almost automated. I mean, these are done typically by contract research organizations. You choose your best candidate after you see which one has passed these various hurdles, and you then conduct animal studies. And again, there's a select number of behavioral type paradigms that one would look at, depending on the indication. And once you've done that and you see that you've identified your best lead candidate, you can file for an IND application with the FDA and begin to conduct a clinical trial. Gideon might want to add something to that because he's been through this process a number of times.
Yes. Yeah, so we're very fortunate in our experience with having filed many INDs, and also the industry has evolved now so that there are many, many contract research organizations that will handle the entire package for you. So once we've discovered our lead molecule, as Alan says, that does everything we want, that's safe and selective and in animal models and in the pharmacology of the 5-HT2 receptor selectivity, we're essentially well positioned, and that's a well-defined process to then take it to a CRO that conducts the IND enabling studies, which is the gateway to clinical entry or first in man. I think the key, what we have here is with this really revolutionary new drug mechanism where you don't take a pill every day. You essentially, with a psychedelic, are talking about a drug you only have to take potentially once a month, or it might be more frequent depending on the indication. So what that means is the clinical development, when we enter clinical trials in patients, after you first demonstrate safety in phase 1, where that means you dose to healthy volunteer and determine what dose essentially is effective, or you get a psychedelic effect. the transition is very smooth then to a patient population, because unlike many once-a-day pills or an SSRI, that can take up to 3 months to work. And the real revolution is basically this is a immediate onset kind of action with psychedelics, where you can get a readout for the disease relatively quickly, even with a single dose, which is what's going on now. All the first generation psychedelics are pretty much being dosed, give it once and see if it works. So in the long term, you're certainly going to need to dose more frequently and figure that out. And to that end, you're going to want a safe drug that you can dose on a relatively frequent schedule, maybe once a week, for certain diseases where, once a month or once a year really isn't going to be the standard treatment for a disease. That may work in specific cases as a life-changing experience, but I don't think we can count on that for psychedelics and this mechanism generally. So that's where we're really blessed with having the leading experts who are working on these first-generation drugs and clinicians. So the person we're working with is conducting the initial trials with psilocybin. And so we'll be able to benefit from that experience and see the pros and cons, what's the dose, what to look for, so that we can follow on relatively quickly and basically draft off of all the experience with the first generation clinical data in depression, psilocybin, or cluster headache, and then use that knowledge to really efficiently displace those compounds with the second generation safer drug.
I guess I would add also, Gideon, I guess one of the reasons that one shouldn't be using the mushrooms themselves is that you really don't know the amount of psilocybin that's going to be present in different batches of these magic mushrooms. And, it probably depends on the time of year, how much of the active ingredient there is there, where you gather them from. And also, I think there's a danger in that one doesn't know, with different individuals, what the rates of metabolism is, what kind of metabolizing enzymes are present, so that a small amount of the mushroom may cause them to have a really bad trip. Whereas in other cases, other patients may be able to ingest 3 grams of the mushroom material and do okay.
Right. Basically, we take the guesswork out of the dosing, essentially, and that's key, because if you don't take the guesswork out, then you've got 2 different variables you're trying to optimize in parallel, which is extremely difficult. So essentially, we're creating homogeneity in the procedure, so we have essentially a goof-proof or a foolproof treatment where we know the dose, we know the duration, we tailor it to the patient, we have precise different doses for each patient that we can basically optimize and know exactly, what that is and what the response is. So, again, we're sort of taking the sad thing is sort of taking the magic out of the magic mushrooms.
That's a good way to put it.
And making it, a straightforward, routine, foolproof treatment.
Yeah, very helpful. And I think it's worth mentioning. So we have cluster headache as a program that we have going on. Obesity, we have a number of targets that are ready to go through Alan and Brian Ross' prior work. Also looking at the areas of addiction. Alan, do you want to touch on that briefly?
Well, there we've contacted Peter Hendricks at the University of Alabama. He's already actually studying cocaine addicts, treating them with psilocybin. And he told us last week that in the case where he unblinded part of that clinical study, the first 10 patients were actually showing some very promising results with taking psilocybin. So we're optimistic that in working with him in the future, with our improved new chemical entities, that we're going to do even better. So I think this is a tremendous application to cocaine addiction, but also there's a potential for using this, and that's alluded to in the literature, one way or another, to smoking cessation, as well as to decreasing intake of alcohol.
Perfect. And I think just lastly, you've seen the three co-founders here, but we do have considerable amount of other colleagues that are not online today, and the team is growing. We will be adding some pretty senior members in short order. So it's a company that has been around for a while and is really accelerating into the next phase. Hey, Merrick, if we have any time, I'd love to take questions. If not, thank you.
Yeah, just one question from the audience. Thank you so much for the talk, Ian. If polypharmacology is a keystone concept for the effects of psychedelic drugs, how can we create a molecular model for drug screening? This departs from the classic single target specific indication model in drug discovery.
Yeah, so regarding the polypharmacology, there are a number of, I don't really have time to answer that in detail, but we've taken that into consideration. For example, the 1A receptor site generally washes out some of the psychedelic activity. The 7A receptor site is somewhat interesting, but really the focus should be on the 2A receptor site. And there's a lot of other targets there that are quite interesting. 1A, for example, is quite interesting for an anxiety drug. But sorry, I don't think we have time to really get into the nuts and bolts of that. But
yeah, I'll let people know to reach out to you as well. Feel free to leave your email in the chat. Thank you so much, Ian. We really appreciate your time. Thanks for joining us and looking forward to chatting soon.
Appreciate it. Thanks.
Yep. Bye-bye.